welcome everyone to this FIDELIO webinar. I'm Andrea Burden, and I'm very honored to have with me today Dr. Peter Vestergaard as a co-presenter. So for the webinar, of course, if you have questions throughout it, please feel free to unmute yourself and interrupt us. You can also enter your comments into the chat, and I'll do my best to monitor that while presenting. Otherwise, there will be time at the end for general discussion. And of course, if you're comfortable, please feel free to put on your camera so we have a nice virtual audience. So today we're going to talk about register-based research, getting more into observational healthcare research or epidemiology, with the aim of discussing how we can go from bench to bedside research, really with how healthcare data can not only improve the line of research from the bench, but also feed back in to stimulate new research ideas. Oh, for the agenda today, we're going to first, I will go over what are healthcare registries and what are the common study designs and how can we use this type of data to answer specific questions, particularly related to diabetes and bone. And then Peter will take over and discuss how we integrate this with basic research. So we'll start with the first part. So the goal of medical research, well, generally what we're trying to do is always identify the effect of some form of exposure, which in often cases is a medication, but this could be many factors such as environmental aspects, other diseases, on an outcome. When we think about medications, we generally know that there is a long process of drug development, and many of you may actually be working in the drug discovery, preclinical research, or even within clinical trials. Once a medication makes its way through this whole process, it ends up being approved, hopefully by one of the main governing bodies. And then the healthcare registers or epidemiologic research comes over here in the phase for post-marketing research. Now we know that the randomized controlled trials, those aspects that you saw in the middle, are really the highest level of evidence when we're thinking about causality, so the effect of a medication on an outcome or the effect of any intervention on an outcome. However, there are some problems that can occur within clinical trials that require the phase 4 research to be done. And that is that these trials are often in very controlled environments. They have very restricted patient populations. And of course, for some questions, they're expensive, time-consuming, and potentially even unethical. So this is really where this epidemiologic research can come in. We can begin to fill the gaps of the randomized trials, and we're going to actually expand a lot on this in the course that you'll be a part of next week. So how do we actually do this observational research? And this is really the crux of the talk today. Well, we use routinely collected healthcare data and we apply observational study designs. So this is that idea of healthcare registers that we started off with. And what is it to be observational? Well, it means that we're using data that was not generally collected for research purposes. So for many of you in the labs, you're going to analyze data that you yourself collected and was based on a protocol that was written to ensure that you have all the variables you need and the timing of those variables to answer the research question that you proposed. For us, when we're using healthcare registers, we're often using data that was actually collected for purposes such as reimbursement, so health insurance data, general surveillance, so digital healthcare records, or things like the census data. And this adds into an element of complexity because we may not have all the aspects of data collection that we would want to answer our question, and we have to ensure that we match our research question to the database that we have available to us. But the big advantage to them is that they are extremely large population-based data of anonymized patient information, and this does give a lot of power to look at specific questions, particularly ones on rare adverse events. Now, I'm a visual person, so when we're talking about healthcare data, I think it's sometimes helpful to give an idea of what it is that we're actually looking at. And for this, we could imagine that we had a patient and she's going to go on a journey through the healthcare system. So this may start by her going to her healthcare provider, And we're going to have some basic information on her vital statistics, maybe her age, also, of course, whether she's alive or has died, and as well as her gender. At her healthcare provider, she may get some information about a basic diagnosis, some new healthcare concerns, which then lead her to the pharmacy to get some medications, which we have information on. Unfortunately, this patient may take a fall and have a broken hip, which leads her to the hospital where she ends up having a surgery to have a hip replacement. At the hospital, she'll see a specialist such as an orthopedic surgeon, and potentially she may end up in a specific disease registry where we're monitoring those who have hip replacements. Luckily, she survives this healthcare event, ends up returning back to her home where she continues to get different medications and continues to have follow-up visits with her primary care doctor. So these are the types of pieces of information we're talking about when we talk about observational healthcare registers. So we often have information on medications. This could come from the pharmacy dispensing or even the physician prescription notes. We have information on the hospitalizations, the outpatient and admission data, primary care and specialist information. And as I said, there could be these additional disease or medication specific registries and their vital statistics. But as you can get from the first picture, it's not just that we want each of these individual pieces when we're doing research, but we want all of these individual parts to come together into one unified source that's linked based on a patient identifier. And once we have something like this, we can really begin to build a picture of a patient's experience over time and allows us to better anticipate or study the causality between an exposure and an event. But as you may have guessed, we don't want this for just one patient, we want it for an entire population. So this could be an entire country, such as the population of Denmark, where they have really wonderful data, as Peter knows, or Germany, Canada, many countries have such large population databases. Additionally, we want to make it anonymized. Naturally, this is very sensitive data, so this is held with the utmost of care. So every patient is anonymized, and then this allows us to be able to develop the analyses that you'll see throughout the talk today. When we talk about different type of registries or healthcare data, it can come in many different forms. We can have basic things such as pharmacovigilance data, which is kind of this early signal detection of case reports when a new concern arises about a medication. We can have what we call administrative claims data, which gives us information on whether or not there is an exchange of monies. This is typically healthcare insurance data, electronic medical records, and also the patient registry. So when you think about it, you can think pharmacovigilance is that safety signal, then you have money, now we have the doctor's notes, and then finally some more specific information about a specific medication. The main message, of course, is always to remember that none of these databases were really collected by researchers for the specific research question that we are aiming to answer. So now when we, sorry about that, we advanced quite a bit. So now when we talk about observational study designs, what do we mean here? Again, unlike what we often do in the labs, we're not applying any experimental intervention to our research. We have instead information that is naturally occurring in a healthcare environment, and we are observing what happens and applying different study designs and analytic techniques to be able to determine the causality. So the typical flow chart that you can see on observational study design starts with that question: did you assign the exposure or not? If the answer is yes, you're generally doing an experimental design, and this may be what many of you are doing in your research projects. For us in the observational world, the answer is no, and then we can either go through a descriptive analysis where we want to just characterize what is going on, or an analytic study where we try to assess the relationship between the exposure and outcome. And for these, there's generally three main types of study designs. The first is the most basic, which is a cross-sectional, and this means that we are looking at the exposure and the outcome at the same point in time. So this could be something like a survey data, where we gather information on what medications a patient is taking, as well as specific healthcare events, all at one point in time. The next one would be a case control study where we actually see who has an outcome of interest. So who might have a fracture versus who does not. And then we can look back in that patient's history to identify if they got different types of medications. The most common study type is the cohort study, and this is designed very similar to a clinical trial where we actually start with those who get drug A or drug B, and we look forward in their history to see if they end up having the outcome of interest. And we can also have this done in the future where we look at a prospective cohort study where we recruit patients into our study, look at what medications they're getting, and follow them forward into the future to identify their outcomes. So this was a very quick crash course into epidemiology, healthcare data and study designs. We'll go into this into a bit more detail next week. But for the next part, I wanted to very briefly go into what kind of questions can we actually answer with this type of data and these types of study designs. The first most simple aspect is to characterize. So to better understand what's going on in the environment, these are things looking at the incidence and prevalence of disease, and they're really useful for developing new hypotheses and identifying where there's need. The second aspect is to assess the causality, to identify if there is indeed a relationship between an exposure and an outcome. And finally, naturally, what we all would like to get to, and this gets us closer to personalized medicine, is the idea of prediction. So can we actually develop statistical models with our healthcare data that would improve our ability to predict which patients will have a specific outcome? So when we think about characterization, this was a really nice study and actually used great data visualization techniques to characterize treatment pathways in type 2 diabetic patients. What the researchers did is they used 3 different databases, one from the US, one from the UK, and one from Japan. And they were able to then combine all of them into one model and assess what patients received during their trajectory in type 2 diabetes. So the inner circle is the first line therapy, so the first medication a patient received. The second circle is the second medication they received, the third medication, and the outer is the fourth. And so what we can see is that generally speaking, we see patients receive metformin as their first line therapy, which would be expected based on the current clinical guidelines. However, what they start to receive as second line therapy becomes quite heterogeneous. We see quite a broad mix of where they go. And of course, it becomes more and more diverse as we go to the third and fourth line therapies. And naturally, we could see really important differences when we look at different countries. And this helps us understand that when we're trying to do research, where can we actually generalize our findings to? So for example, we can see that the US and the UK have a moderately similar first line therapy approach, with metformin being the main use. However, when we go to Japan, it's quite a different story where there's not a dominant first line therapy. So this is quite important also when you're reading other research papers to understand the healthcare system in the individual countries. The next step that we would want to take is, after we've kind of developed an overall view of what's going on with the patient population, maybe to actually assess the causality between diabetes and fracture risk, or medication and fracture risk. So this was a study done a few years ago where they actually went to look at whether or not diabetes was truly a risk factor for fracture. And on the left-hand side, we can see their figure where the solid line are the non-diabetic patients, the dotted line are the diabetic patients, and on the y-axis, we can see the fraction without a major fracture. So over time, we can identify that the diabetic patients had a higher fracture risk as they dropped off more frequently. So there were fewer patients without a fracture over time compared to the non-diabetics. When we look at the overall, sorry about that, my mouse is very sensitive today. When we look at the overall adjusted risk factors, we could also see that when adjusted for the normal FRAX risk factors or FRAX probability, diabetes came out as a significant independent risk factor for having a major osteoporotic fracture, and this continued regardless of the age, so even in those under 65 or over 65. In terms of prediction, this paper on the left-hand side that we just looked at before went to see if the factors could actually predict fracture in diabetic patients and non-diabetic patients. The diagonal line here would be the predicted value, and we could see that for the non-diabetics, the FRAX score actually did a pretty good job at predicting fracture. However, in the diabetic patients, it underestimated the fracture risk, suggesting that there is more to fractures in diabetics than what we would normally predict with our FRAX variables. Another study then took this one step further and added in to look at whether or not the trabecular bone score could be an additional predictive tool. And as we can see in the blue line, that overall the trabecular bone score was a better predictor of fracture risk in diabetic patients. So this leads to a lot of the work that we're doing here within the FIDELIO cohort to try to better understand why is it that these patients are fracturing, and we're starting to understand more and more that they are not similar to our traditional non-diabetic patients. And with that, I'm then going to hand the floor over to Peter, where he can then take it one step further and say how we can use the type of research, particularly causality research, to integrate this with basic findings as well as spurring on future research.
Thank you, Andrea, for a very nice talk. I thought I'll start with both the big picture and the very small picture. If you go down the bottom of the screen, you can continue all the way down to the atoms and the quarks and the whatever elementary fundamental particles. However, what is captured within this picture is the frame where we as biomedical researchers or healthcare researchers usually operate. So if you have, for example, a single cell, you know how it operates. However, if you pool multiple cells together, they start to crosstalk, and you may have regulatory mechanisms that you don't anticipate from studies on single cells. So when you move up through the picture, you come to the tissue where the cells crosstalk. They may have a lot of regulatory factors, endocrine regulators, and so on. So then you need to consider which methods you need to use. Now we heard about the registers, what they can contribute with, and I'm going to elaborate a little bit on the biomechanical competence of bone a little later. We may then move up through the clinical physiology. How do we expect bone to behave? How do we expect the number of fractures to be? Then we may want to intervene to say lower the number of fractures. Therefore, we would of course need the randomized controlled trial as presented before. However, the randomized controlled trials have limits in duration. There may be certain side effects or good effects that are only seen with very long observation periods that we can only cover with epidemiology. Certain very rare events can only be captured if you have very large samples that are not feasible with, say, a randomized controlled trial, or there may be some subgroups that you're not able to study, say, differences between men and women, old and young, and so on. So this leads up through the picture through clinical epidemiology, register research through epidemiology, and as my old teacher once said, you can continue all the way up through the universe and to the edge of that, but let's leave it with what's practical at the moment. Next slide. I've agreed with Andrea that you try to advance the slides, then we'll do it old-fashioned. Now I thought I'll take a little bit of an odd example first, showing how you may sometimes stumble upon signaling pathways using just an example of what Andrea just showed us. Usually what we are told is that if you have a drug, it's a risk factor. That is, you have an increased risk of something, in this case, breaking your bones. So if we take something completely different, if we take the psychiatric diseases, however, most drug users within the field of psychiatry, they have, as we usually see, an increased risk of fractures. This could be if you have manic depressive disorder and you use, say, anti-dopaminergic drugs to treat, say, schizophrenia, mania, and so on, they increase the prolactin levels. Prolactin suppresses estrogen and also testosterone for that level, and you get a state that sort of mimics the postmenopausal state, or at least something like that. Therefore, when you don't have estrogen and testosterone, your BMD, your bone mineral density, decreases, and you expect to have an increased fracture risk. However, there are some differences. If we go to the antidepressants, those receiving the tricyclic antidepressants, they have normal BMD. That is, if you scan them, it seems okay. Their bones are dense as they should be, or have the density that you expect. However, they have more fractures. Then, as Andrea pointed out, we may think about, why is there this discrepancy? Why do they have more fractures? In this case, it could be the effects on balance on the cardiovascular system. That is, you have a decreased postural balance, you're more likely to fall, to tumble and break your bones, although they may be sound if you scan them. Going further down, if we have the more commonly used, the selective serotonin reuptake inhibitors, they can interfere with serotonin and affect the bone mineral density. They may also affect your postural stability, but here you have two different factors that may work together. This was just to set the scene. Next slide, please. Then, just to present some of the research that we did. Usually, as I said, you expect more fractures. It's very rare that you find a drug that seems protective against fractures. However, if you do enough studies, by chance you can find a drug, just because there's 5% significance limit, that there are some who decreases fracture risk. However, this study that we did, and also subsequent studies, have indicated if you use lithium, which may be used as a mood stabilizer, if you have very alternating mood, you may be either manic, that is overactive, or depressive, then you can use this to stabilize the mood so that you have a normal mood level, that you're not depressed and not overactive. Up the ordinate, you have the odds ratio of any fracture in the first part of the slides, and if you're around 1, you have the same expected risk of breaking your bones as a background population. Then it seemed for the any fractures in the left-hand panel of the figure, that the more lithium, that is the accumulated dose down below on the abscissa, the less fractures you had. Actually, if you had used more than 850 defined daily doses, or at least around a little bit more than 2 years, if you used 1 defined daily dose or 1 dose every day, you had fewer fractures. Hip fractures, not really any significance. However, for the collar fractures, although there was an increased risk with low cumulative use, there's a decreased risk with high cumulative risk. And the same was seen for the spine fractures. So there's a decreased fracture risk. It seems to be somewhat dose dependent. So of course you can think, is this just a coincidence? Is this just by chance that we stumble upon that? Should we forget about it? Next slide, please. However, if you look a little bit deeper into lithium, one of the things it can do, at least in a petri dish, is that it can affect the Wnt signaling pathway. And of course, in the state where we are with diabetes, the insulin receptor and also glucose transporter may actually interfere with the Wnt signaling. And here on the A panel in the left side, we have the absence of signaling in the unliganded states. And over on the right side, we have the signaling with the Wnt ligand bounds. So what this leads to in the lower right half of the picture, that is, if you have a signaling via LRP4 or 5, then you have an increased osteoblast stimulation and an increased bone formation. If you signal via LRP5, via LRP6, you inhibit or may inhibit osteoclast stimulation and decrease bone resorption. This, of course, is interesting. Now, just put this up in your memory. This could, of course, be of interest with the lithium. However, LRP is a lipoprotein receptor. So could there be something, if we remember that until a little bit later, what about if we lower cholesterol too much? Because that's the LDL cholesterol in particular, which is one of the stimulating factors. Next slide. This is from an in vitro study where you've made a knockout model. Up in panel A, you have shown, first we have these 6 weeks of initiation, then we initiate treatment, and then we analyze this mouse model. They removed some bone from it. It's a knockout where you removed LRP5. You may either treat with a vehicle that is saline, which is supposed to have no effect. You may also use something LP25, which is supposed to bypass what you knocked out, or you may cultivate this with lithium chloride added, which is the striped ones. In panel B, and I won't go into much detail with this, out on the left side, you have the bone volume from histomorphometry versus total volume. The first bar is if you only give the vehicle, you have sort of blocked, you don't have the LRP5 anymore. So you have very low bone volume, you have osteoporosis. Then if you cancel that out, well, you increase your bone. But the same happens with lithium, the third bar there, and sort of the same way all across. So it seems that perhaps with lithium, if we go to the cellular level, we may find something which mimics what we see on the population level, leading us to say, well, now we are one step further in these Hill criteria for causation. Perhaps what we do see on a population level mimics what we should see on the cellular level. Then we, of course, need everything in between. How do the bones behave in HRP-QCT, DEXA, and so on? How do the turnover markers behave, and so on? But again, this could point us in the direction that perhaps we have something working on the Wnt signaling, a pathway that's effective. And of course, we now have also newer drugs coming onto the market which affect this pathway. It's just an example of how you may, if you screen a large number of drugs in these registers, you may come up with ideas for potential candidates, perhaps not the drug itself, but the pathway that it interacts with that you can use to create potentially good drugs for the bone. Next slide, please. So now back to the diabetes. Sorry for the digression, just to show you an example of how we can do this. Morten Frost excellently pointed out in his webinar, just to boil everything down, if you have type 1 diabetes, in theory you have thin bones, therefore you're expecting more fractures. That is also what you do see. However, there's a discrepancy between the number of fractures you see and the decrease in bone density. So there's something we don't capture with traditional scanning techniques. Going into type 2 diabetes, the patients actually have denser bones, so we should expect lesser fractures because the bones seem to be good. However, we don't see fewer fractures, we may actually see more fractures, leading us to think, is this a matter of bone biomechanical competence or something else perhaps false? Next slide, please. Now, we can use the registers to answer some of these questions. For example, is it actually true that we find, as Andrea pointed out, more fractures with diabetes? Could this be that if you're a diabetic, well, I hit myself, maybe I should just go home and put some ointment on it, then it'll heal. Now I have diabetes, so I should go to the hospital and see the GP, and then it's put into the registers. It could be a Bergson bias. If you have another disease, you're more likely to have occurrences of fractures registered. So we could go in to see if that's actually correct. Then we can also use the next level of what I pointed out in the prior slide with all the levels from the big world down to the individual atoms. We could use what we know from DEXA scans. As I said, does this mimic what we see? It doesn't in type 2, it doesn't in type 1 completely. We could move one level up with the HRP-QCT. This is something that the DEXAs don't capture. As pointed out by Andrea with the TBS, for example, there could be some things because DEXA is a two-dimensional scan. However, and this leads me to what I'm going to talk about on the next slides, what about the bone biomechanical competence? Because whenever you use an X-ray technique, that is DEXA, HRP-QCT, and so on, in theory, what you do capture is a mineralized matrix. You don't capture the unmineralized matrix. So if we go to the next line, we could look into the bone biomechanical competence. For example, trying to bend the bones. Do we find what we expect? And I can see it seems to advance, but that's okay. Because my point is, and I usually use this as an example, if you take my finger, it has about the same dimension as a piece of chalk. The chalk I can pretty easily fracture. If I put it in a DEXA machine, it would tell me this is excellent bone. Probably an HRP-QCT machine would say this is really, really good bone because it's very dense. However, if you put two different compounds together, something which is very strong and something which is flexible, then you get a composite material which is lightweight and stronger than the individual components. And the analogy I'm giving here is sort of if you want to build a building, now we can build more than 800 meters into the sky with the Burj Al Arab or Burj Al Dubai. And the only reason why we can build so high is so we combine rebar, that is steel, within the concrete. Although we may think of steel as being very strong, these fibers that they are within the concrete makes it lightweight and yet very strong. And this is also what we see with bone. Next slide, please. Now, because within the bone, the rebar is collagen, analogy is collagen. And collagen has these fibrils, which are sort of spun together like a thread. And if you unwind them, then you have osteogenesis imperfecta. That's how that occurs, because then it doesn't bind the way it's supposed to. Next slide, please. Now, if you think about the rope that I just showed you, this has sort of a sawtooth shape, and this gives a very good capability to interlock with the crystals. So it has a good pull strength and a good ground for tendons to interlock into the matrix. And the teeth in the saw, they grab these hydroxyapatite crystals, yielding a very strong compound. And the ragged form acts like a spring, so you also have good elasticity and good compressive strength. So with the calcium, you only had a very rigid and thus very frail structure. But if you combine these two, you have something which is even stronger than the individual compounds and is not bendable as the collagen would be alone. So if you have diabetes and have a mismatch between the soft matrix and the hard matrix, that could perhaps explain some of the discrepancies we see with the fractures. Next slide, please. This is just an illustration that sort of think about the collagen fibers as this rope that I have drawn there. Think about the hydroxyapatite or the calcium phosphate crystals as these hexagonal crystals which sort of interlock with the collagen fibrils. And with each hydroxyapatite crystal is hexagonal, and it consists of these three rhombi, and each rhombi consists of two triangles. And this again gives a very high both pull and pressure strengths. So now we may be looking into, if we go down the pyramid, there could be something about the bone biomechanical competence that you, from the basic research side, could contribute to explain why there is this increased fracture risk, although the bones, perhaps even in type 2 diabetes, may look good if we put them in a DEXA scanner. Next slide, please. So one of these rhombi contains one hydroxyapatite molecule with this chemical formula. And also further just to the structure, if you have more water, more hydrogen oxide in it, then you have more crosslinks, that is, which contributes to the strength also. And with age, the hydration of the crystals may go down, so they more look like a frail old branch that breaks or shatters, whereas in youth, the bone may look more like a young branch, which is filled with bending strength, so you can bend it sort of if you imagine that. Next slide, please. So, going a little bit back, now I've gone down the ladder into how the bone is organized. We could ask the question, is it mirrored in BMD? No, not completely. If we take the DEXA scans, which we can also get from the registers, could it have something to do with the competence of the bone? Perhaps. Again, however, although we may top all of these things on top of each other, it doesn't completely explain the discrepancy between what we measure on the organ level with what we see on the fracture level. Therefore, there is a dark space which we don't understand. And one of the last factors could be if the patients fall. They may be more likely to fracture their bones, although these are of normal strengths. Why do you then go and fall? Well, if you have a low blood glucose level, then you may become dizzy. You may fall over. If you're in a car, you may be in a car accident. We can also go into the registers and examine that. Did they have hypoglycemia episodes that led at least to a hospital contact prior to the fractures? Yes, it explains some, but only a minor proportion against of the increased fracture risk. So some of it is hypoglycemia, although not all of it. Some of it is poor bone strength. So taking this a little bit further, if we then intervene with the drugs, Andrea showed this nice trajectory of, well, you may start with one drug, progress to the next for one reason or another. And some of these drugs, or as I mentioned before, may interact with bone signaling. So one question is, if you have diabetes, you have an increased glucose level. The glucose may interact with the collagen that I showed earlier on. The crosslinks, those of you who try to put sugar into a pan, you know you can destroy it by that. And the same can happen with these collagen fibrils. If you have glycation of them, they don't work the way they're supposed to. They don't adhere in the bone. So one natural question would be, well, there's increased glucose level. If we lower it, does that improve fracture risk? Is this drug dependent? There are some studies with the GLP-1 agonist suggesting that some types of these who are more like a natural glucagon-like peptide than others are less detrimental if they look like GLP than if they don't look so much like native GLP. What about if we use biosimilar drugs? Could we sort of think the next step, and Andrea is going to come a little bit back to that later on when I finish talking. If, for example, we now know that this twerk on the molecule gives the effect, this twerk gives the side effects, and this is just a binding site, now we know something. Could we remove one of these and then perhaps get the good effects with fewer side effects and still maintain the ability to bind, for example? How do other drugs, besides those using to lower glucose level, how do they work? For example, if you lower blood pressure, that can be good for some things. However, it can also be too low. You become dizzy, and you fall, as I mentioned. Again, in diabetes, you don't want too much cholesterol. It can be bad to your arteries, to your heart, to the risk of clots in your brain and so on. So you want to lower your cholesterol levels. However, these may also interfere with the LRP signaling that I talked about, because statins may interact with the mevalonate pathway, which is prevalent in bone. So is there something with statins, if we lower LDL too much, is this a risk factor? This is also something that we can elucidate using the registers, which would be very hard to do using just the RCTs. Next slide, please. And that finishes all of the questions that I sort of put up into the air, and I'll leave the rest of the floor to you, Andrea, to complete the circle, so to speak.
Thank you very much, Peter. So what we wanted to do primarily with this webinar was kind of probably create more questions than answers, as that's typically where we are with the relationship between diabetes and bone. But I think whenever we talk about particularly the relationship between medications and an outcome, it always kind of comes back to this picture, and it looks like it's a one-directional factor. So we start off by this aspect of, is there a biological connection between what we're looking for, then taking it to preclinical studies, hopefully getting it through a trial. But the reality is we don't actually look at fracture outcomes very often, or these medications on reducing fractures, with the exception of bisphosphonates for osteoporosis. So even though when we're doing studies out here on the far right-hand side, looking at, is there a relationship, we have learned from all the research that comes before. However, I think an important aspect is we can also find these unusual patterns of medications being associated with an outcome, for example, lithium and fracture, which then sends us back into the early phases of research, going back to the bench and trying to explain what we're seeing in clinical practice. So it is a continual loop, and I'm sure many of you are realizing this now. It's a very diverse group of researchers that we have here in the FIDELIO consortium, bringing together all phases of clinical research. And I think it's really important, and why I really like the FIDELIO consortium, because we can learn from each other. So for example, the work that some of us are doing on trying to look into our healthcare register data and not only test hypotheses, are certain drugs or certain lab values associated with fractures, but we can also start to develop and drive hypotheses of maybe new exposure outcome relationships that we hadn't seen, that will then come back to some of you working in the labs to try to explain if there is a logical connection there. So with that, as I said, it's a bit of an introduction. Next week, we are going to, in the course, talk a lot about how we do this, and also answering some of those questions that Peter alluded to regarding bias. I'm sure many of you have heard about that there can be this problem of bias with observational studies, and how do we trust the data that we're doing or that we're working with. And so we will address these questions quite a lot so that you can better assess the clinical research, so that you can inform your laboratory work. As Peter said, this aspect with statins is a hot topic, as well as things like the GLP-1 inhibitors. So we will go through a lot of these topics. How do we know if someone used the right database to answer a question? Did they use the right study design? And did they have any potential confounding or other forms of bias within their data set? So with that, I think we've left lots of time for question. We're about 1 minute early even. So I will open the floor. Feel free to ask us any question that you have. Perhaps if there's something in your research that you want to investigate and see if it's observed in a register, we can tell you more about the data that we have available. But yeah, any questions are welcome.